Welcome everybody to today's LA Biome City mini reviews webinar series. My name is Yvette van Eenennaam and I'm the General Manager of LA Biome City, the Health and Medical Innovation District here in the heart of Adelaide. And I'm very pleased to be your chair of today. And before I introduce the presenters of today, I would like to acknowledge the Kaurna people, the original custodians of the land we meet on today. And I'd like to pay my respect to the elders, both past, present, and future. So if we look at those weekly short webinars, they're organized to really inform our South Australian researchers, clinicians, students, entrepreneurs, and business people about the key capabilities in our innovation district, but also to spark ideas for further collaboration and new partnerships. And I also welcome our national and global attendees who are keen to seek opportunities with companies and researchers, clinicians residing in our innovation district. So today's session is being recorded and also will be available at our website, LA Biome City website, later this week. So we have all our past webinar recordings on the website. It's containing quite a diverse and extensive range of capabilities. So if you're interested in specific areas and like to explore further capabilities, please visit the website. I would also like to mention the Q&A option. It's at the bottom of your Zoom window. So please post any question you have during the presentation. I will have a look at those questions and I will ask the presenters at the end of the presentation. So this afternoon, today, we're very fortunate to have, again, two great speakers from an Adelaide-based company, which are currently distributing their invention to many places across the globe. So today we're joined by Jack Brown and Rhys Hill from LBT Innovations. So LBT is a medical technology company that is commercializing their novel artificial intelligence-driven diagnostic technology for clinical microbiology. It's also known as APAS Independence. Jack has spent over 10 years working in different corporate development roles, mainly focusing on identifying new commercial partnerships for technology developments. And Rhys is LBT's research director and inventor of the LBT APAS technology. So he has been working in image processing and machine learning for, I believe, more than 20 years. And prior to joining LBT, he was a researcher at ACVT, so working on the implementation of a range of different computer vision technologies. So I'd like to hand over to, I believe, Jack at the start, and I'm looking forward to hear the presentation and your capabilities.
Thank you, Yvette, and thanks for the opportunity to present today. It's great to have a chance to share what we're doing with the wider Adelaide community. And so first of all, I'll provide a bit of a background on the company, and then I'll hand over to Rhys to dive a bit deeper into the technology. He's much more qualified in that respect than myself. So LBT, we're a public listed company on the Australian Stock Exchange. We're in the early stage of commercial launch of our image analysis technology, the APAS Independence. Over the last 3 years, we've really focused on building out our capability within the company, and we've built a team of around 30 people based here in Adelaide, really in the core areas of artificial intelligence, software engineering, and clinical microbiology. We've got a PC2 level lab that we have based here in the city, and all of that is underpinned by our regulatory and quality management systems that enable us to develop technology that's for healthcare applications, so able to meet those quite stringent regulatory requirements. So a bit about our company history. We started in 2004 and we were formed on a technology called the microstreak, which came out of the assay pathology. This was a device for automatically streaking a specimen onto an agar plate. And we sold that in a partnership with the French diagnostic company bioMérieux from 2009 to 2015, and sold around 500 of those systems worldwide until we eventually divested that technology in 2017. During that time, we then focused on, well, what would be the next technology for the company? And we looked at the next bottleneck in that culture reading process. And that turned our attention to the plate reading aspect. And that's when we partnered with the Australian Centre for Visual Technologies, now known as the AIML, and Rhys, really joined the company at that point. And that's where we developed our automated plate assessment system, or APAS. And the little black box you can see there is one of the early prototype devices that we actually used to gain our first FDA clearance in 2016. This was a manual loading device, and once we had that clearance for the manual loading device, we then spent the money on developing the fully kind of commercial product that you see there on the right, which is called the APAS Independence. And so we're in this is launched globally in Australia, the EU and the US. We've got partnerships with Beckman Coulter to sell it in the EU, and Thermo Fisher more recently to sell it in the US. And we're also developing a pipeline of applications that we can then run and process through this system. So we can process urine specimens and MRSA specimens, and we're working on developing the technology for additional applications such as VRE infection control and a antimicrobial susceptibility application as well. So our technology development's always been very much, I guess, market driven and focused on, well, what is the need of our customers? So when we look at the culture reading process, there's about 2.4 billion plates read around the world per annum, and it's a highly inefficient use of resources. Over 85% of these plates can typically be negative, showing no sign of infection for the patient, which is really great news for the patient, but it's not a great use of a really highly skilled resource, being the microbiologist that reads these plates. And this resource is in short supply. There's a vacancy rate of 9% in the US, which means that roughly 1 in 10 positions are vacant at any one time. So what you have is this, really manual process requiring a really highly skilled resource, and it's not a great use of their time. So it's a really ripe application for automation. And that's where we developed the APAS Independence, which you see on the right. So the real smarts of this device that Rhys will tell you a bit more about shortly is in the middle. That's the image analysis AI system, which is able to read different specimen and media types. And then all around that is the kind of automation functionality that we've added on that delivers the efficiencies required by our customers. So we've gone through, we've got patents protecting the technology. We've proven it in large scale clinical trials. And we've got the global regulatory clearances. So we've got FDA clearance, we've got CE mark and TGA approval, and we've launched the product with sales, as I mentioned before. So taking a new technology to market, really means you have to get the early adopters and innovators on board. So we work with St. Vincent's in Melbourne, we work with Hennepin Health and Labor in the US, and Labor Dr. Wisplinghoff. We've also worked with the likes of Johns Hopkins Hospital. And these are the people who are going out there, and they're really, I guess, telling the world how great our technology is and what it can do for their lab. In the study by Johns Hopkins Hospital, we could actually show that, using our technology, they were able to identify, specimens that they might have missed otherwise just through manual reading. So that's really paved the way and built awareness amongst the industry and paved the way for us to launch the product more widely with our partners Thermo Fisher and Beckman Coulter. So I'll just hand over to Rhys now, who will talk a bit more about the technology.
Thank you, Jack. So there are many companies around that profess to be AI companies. And I guess, I guess they're all pretty similar. So all AI problems kind of follow a very similar series of steps. There's always a data collection step. So you need to collect representative examples of whatever problem it is you're trying to solve. AI generally requires that someone then annotates those with the outcome that you want the AI system to determine. You then have to do something to make the computer go from the input data to the output data you specified, which is the training part. And that's normally where most people stop. And there's a lot of companies now using AI systems solve a range of different problems. I guess LBT is a bit different because we control the entire pipeline from top to bottom. So we have designed and sell the image capture device. So you can't put arbitrary pictures of plates through our system. It won't work. You have to use the system itself. But the benefit you get from that is we have a lot of control over how the photos are taken. We can require very specific types of photos to be given to our AI system. And because it's medical device software, and particularly because it's autonomous medical device software, there's a really high burden of proof on us. So we have to perform a lot of very stringent testing. So to get the system through the FDA clearances, which we'll talk about a bit more in a moment, we had to do a 10,000 patient trial that was spread across 4 different sites in the end, one of which was mostly used for development, but it was a very large scale exercise, and it's quite uncommon in AI companies. I think next slide, please, Jack. So the way APAS performs plate reading is that you take photos like those shown on the left. So the pictures that Jack showed you earlier of the APAS Compact and the APAS Independence, APAS Compact is effectively actually fully contained inside APAS Independence. So Compact is really just the camera and the mechanical equipment to hold the plate. So when we take our photos of the plate's actually suspended in the air. So you'll notice you can't see anything. I mean, if you're not familiar with culture plates, this won't make any sense, but there's nothing you can see holding the plate up. So we have a custom designed plate holding system that is been very carefully designed to have minimal interference with the plate. And the reason for that is really just to make the analysis as easy as possible. So part of the strategy we took was to design the entire system from top to bottom to make the analysis of the growth on the plate as straightforward as possible. So the garbage in, garbage out philosophy applies here. We tried not to have garbage coming in, so we had beautiful things coming out. And on the right-hand side, you can sort of see the benefits you get from that. So we are able to do things that, I guess for a person, these things seem very simple, but for these types of systems, they're quite difficult. So we are able to find all the colonies that are spread around the edge of the plate. So you can see there's little blue colonies all around the outside edge. There are colonies on top of that white square. So the white square is actually the identifying barcode that's attached to the plate that the lab uses to track that particular plate through their workflow. We can correctly ignore the dot matrix and the pen writing on the back of the plate. And those are really important aspects of making the system actually work in a real lab, because these are real things that real people do when they're really doing their work. But it's because we design all the parts of the system to kind of work together that we're able to do that. So it's really important for us to have full control over the whole system. Next slide, please. So we thought people might find it interesting to know a bit about what it took to get APAS through the FDA process. So APAS is quite interesting because it didn't have a predicate in regulatory speak. And what that means is there was no equivalent product available. So in most cases, when you go through the FDA, you ideally want there to be something on the market already, which is pretty much the same as what you're trying to do. And that's called a predicate. Now, when you seek clearance to sell something in the US, you would typically look for a predicate which is very similar to what you're trying to sell. You would show that your product works pretty much the same, and then you're allowed to sell your product. So when that's not the case, there's a couple of different options. The one that we took is called a 510K de novo, which is Latin for something new, I believe. But essentially what that means is that there's no law or regulation that covers the device. There's no predicate. So as a result of achieving that clearance, a special section was actually added to the US law that covers APAS in particular. So that 21 CFR part 866.2190 it was specifically added for APAS. So that if you read that, that is very particular to APAS. There's currently no other device in that category. So the first clearance we got gave us that piece of legislation, and that was with Compact. And so once we'd proven the technology was able to deliver acceptable results, we then built the automated system, as Jack pointed out, and we had to do a second 510K for that. But that one was quite simple because that was just showing that the robotic system didn't interfere or cause any kind of poor quality analysis, which was pretty straightforward. The next time we went to FDA though was for MRSA. And this one was really interesting from our perspective because we felt like this one was a lot simpler than the urine. So when you read an MRSA plate, there's a sense in which there's a lot less skill involved. So MRSA plates are screening for the presence of MRSA colonisation on a patient. And the culture plates that you use for this are very clever. So they essentially just change, the colonies that are MRSA positive change colour. And we felt, that's pretty simple. We can get really good performance. It'll be really good. But we had a few, I guess, what we underestimated was the degree of importance FDA would place on these devices working correctly, because MRSA is really critical in a healthcare setting. And so some of although our performance was really, really good, we had a few issues with our testing program that made it a bit difficult to navigate, but we were successful. And so we have another unique to APAS device code for MRSA. So we've got our own piece of legislation and 2 unique device codes. One of the reasons that APAS is different to other systems is it's completely autonomous. So a lot of AI software and systems that you see around is not intended to operate in an autonomous manner. And what that means is that you have to have a human expert essentially vetting all the results. So the machine, a lot of these AI systems essentially work by providing recommendations or scorings or rankings or something similar. But the expectation is that a human operator will inspect the results before they lead to any kind of outcome. And the reason that's done is to de-risk the software, because you're basically putting all the responsibility back on the person who's making the decision as to whether or not the result is acceptable. So APAS is not like that. So APAS does actually make real decisions, and as a result, it's class 2. So if APAS says that a plate is negative, you can throw it in the bin without looking at it. And doesn't seem like a big difference, but from a risk perspective and from the perspective of the regulator, that's a very significant difference. And what being class 2 means is essentially that we have to do a lot of testing and a lot of engineering work. So the burden of proof on us is very high because we allow people to throw plates straight in the bin without looking at them. And so one of the reasons this is important is that APAS is a very high throughput device compared to other AI problems. So if you're talking about like a common case would be looking at, say, chest x-rays or hunting for cancer in an MRI scan or something similar. But those sorts of applications tend to be relatively low throughput. So it makes sense to have a human in the loop because they're highly critical. The number you're trying to do at a time is not that high. Whereas APAS is able to process 200 plates an hour, and having a person vet them all would not be very practical. Next slide, please. So we have some other projects which we're working on. The one that I think we're probably most excited by and is most likely to come out the first is our antimicrobial resistance tool set. So when you're doing antibiotic susceptibility testing, there's a few different methods. The classic method and kind of the gold standard is called Kirby-Bauer distaffusion. So in this method, you create a lawn of an organism where you're trying to work out what drugs would be effective against it. And you place discs on top of it, which you can see in this example image on the right. And you're then looking for how close the growth gets to that disc. And it gives you an indication as to how effective that drug would be in combination with some some various rules and things that are used to make that determination. But we're hoping to add support to this for inside APAS within the next year. And that will again be fully automated. So there have been systems in the past that were able to read these plates, but they've always been one plate at a time, very burdensome on the user to actually make use of the software. So you often have to do lots of things manually to get the result out. But APAS will do it automatically, as with culture plates. The other thing we're looking at is to try and make more use out of the AI system. So normally when we develop our analytical software, we typically have to make sure we only ever operate inside the bounds of what the culture plate manufacturers say you're allowed to do. So when you purchase a culture plate, it will come with a brochure that gives you rules about how you're allowed to use it. That'll tell you when you're allowed to read the plate, how much incubation it needs. But those rules are based on a human reading the plate, not on a machine reading the plate. So we're hoping that the combination of the very reliable color calibrated camera system and the AI will allow us to read the plates earlier than would typically be allowed by a human operator. And we presented some proof of concept data for that at ECMID, and we're working on some future projects to try and get that into the system proper. And we're also always looking at new applications. So a particularly exciting one at the moment is the pharmaceutical market. That is quite different in some ways to our typical product. So our products that we've demonstrated so far are all clinical microbiology. So the pharmaceutical market is a bit different. There's a sense in which it's less regulated and a sense in which it's more regulated. They just sort of have different emphases and different things that they're concerned about. But that's something that we're quite excited by. Next slide, please. And then we have two things that are sort of a bit different. These are not culture plate related. The first one is gram stains, which is a very important tool for microbiology that involves the reading of gram stain slides. So we did some proof of principle work to show that that was possible using the AI system inside APAS, so obviously not the robotic system, because it's the wrong type of robot. And the one that's quite interesting that we were really excited by was WoundView. So WoundView was intended to be a system that could take pictures of sort of long-term wounds, so wounds that refuse to heal. And the idea was that you would take pictures of the wounds and use the software to help track the progress of healing. And I think we all still really want to develop this product, but that one, the technology was really good, but we got stuck because there was no reimbursement pathway, which is one of these things that as a technology person really feels like it shouldn't be a problem, but the reality is someone has to pay. So the current structure of Medicare, and in fact, the same problem is true across the globe. There's just no reimbursement strategy available for something like WoundView. So although we could develop it, we got good results, it seemed very accurate, it seemed like it was really going to be useful. We had partnerships with Professor Fittridge from the QEH. It was all looking great, but there was just the monetization pathway just was really difficult to see a way through. And so that project's been put aside for now until we can resolve a solution to that problem. Next slide, please. I'll hand it back to you.
Yeah. So I guess, just to kind of summarize those last couple of slides, we've got really kind of three avenues that we're focused on from a development perspective at the moment. We've got what I term kind of product extension opportunities. So this is really looking at opportunities to extend the current APAS technology to new applications. So that might be that APAS AMR or early reading application that Rhys was mentioning before, or new hardware systems, for example, kind of incorporating it with the incubator as well. And then we have, I guess, two other areas where we're really looking to try and leverage that internal capability that we've built. So we've been looking at opportunities in microbiology and infectious diseases, so things like the Gram Stain project, where we already have that network of key opinion leaders and partnerships that we can leverage to I guess accelerate the development and taking new technologies in that area to market. And then also AI image analysis applications, where we can use that capability that we've built through developing APAS to target new systems, because I think a lot of people are saying today that they're using AI, but very few are doing it and have been able to take products to market in the way that we have in that respect. I'm quite conscious that we're coming to the end of our time, so I'll hand back to Yvette for Q&A. This is just a little summary slide on kind of everything that we've been through already.
Thank you, Jack. Thank you, Rhys. One of the questions I was keen to hear, but you gave a really nice wide range of opportunities already. Where are the opportunities leveraging your capabilities and technology developed within the APAS, independence? But you kind of reached already from that one view to microbiology and infectious diseases. So thank you for highlighting that, because I think that's of interest to people in our district as well, where you see opportunities for R&D. One of the other questions there is around, going from ideation to development to commercialization. There are many challenges on its way. You will have reached, and you have reached a success with the APAS Independence and several other opportunities on its way. What are the key decisions or companies you collaborated with or people you network that really made it a success? What can you share with the audience in regards to advice when developing technologies or products like yours?
I might take that one. Jack, feel free to jump in. It is very hard. So I only switched to LBT. I reckon I switched in 2016 or 17 or something like that. But I started working on this very late in 2009. So I didn't have a lot of involvement directly with the commercialization, but I was aware of everything that was going on. And it's just really hard to get people's attention. So one of the problems in Australia is the market for this sort of thing is just not very big. So the key markets are really in the US and Europe, depending on what you're selling, obviously. But just trying to get the attention of people who can get it, because you need boots on the ground if you want to sell something in those markets, because people, particularly when you're trying to sell something new, people don't believe it works, which is perfectly reasonable, because you're asking them to trust that your piece of equipment is not going to deliver incorrect results. So this is a big deal for them. You are asking a lot when you put your equipment in their lab. So getting to the point where you can get someone to help you with that, with a name that people trust, just takes a long time. I think our key as you can see my banner behind me. So LBT doesn't actually sell directly. We have a joint venture partnership called Clever Culture Systems. And the other part of that is a company called Headache in Germany. And so they were really instrumental in getting from compact to independent. So they opened a lot of doors, because they're principally an engineering company. So they manufacture centrifuges for a number of different applications. And as a result of that, they had lots of kind of industry links that were really helpful in kind of getting the project moving. But you just have to keep banging on people's doors again and again and again until they believe you. So a lot of it's about getting like a bolus of evidence and like having papers you can point to, and clinical, like our clinical partners are really important, because when you approach Thermo Fisher or Beckman Coulter and you say, oh, we have this great thing, it does X, Y, and Z, they're like, yes, everyone has that. Where's the proof? So you need someone who's willing to stand up for you and say, yes, this really works. So sadly, there's not a silver bullet. And I don't think we're not 100% there yet. So we're still working hard to kind of get some customers' hands, and there's still a bit of a trust issue. So I think you need to get to a certain point where everyone kind of knows someone who's like used it, and then it's all okay. But until then, you really need to just try and get publications out. You need evidence in the field. You need people to kind of go to conferences for you and stand up and say, yes, this works, and just keep doing it until everyone's kind of heard of it and there's trust.
I might just jump in and say the other key aspect to having those early users and feedback is, you're getting insights from your customers, and you need to make sure you're listening to them and taking their feedback about how they want to use the technology, because it's very hard to unwind potential design decisions that you make early on. So really making sure that you listen to your customer base around how you're going to do it, and you're really solving a problem for the customer. There's a lot of great technologies out of there that haven't focused on the customer and end up never getting to market.
Thanks, really helpful. And there's one other question we like to raise as a last question, and that's around, in the slides I've seen that the Australian Institute for Machine Learning has been an important local partner in the R&D space. Who have been other key parties, or key capabilities you're seeking to connect with in the near future, or have been?
So we've worked with a number of parties, I guess over the last kind of 10, 15 years as part of that journey, and AIML have been a really great partner, and to have them local has been very helpful. But we have worked with SA Pathology throughout the development, St Vincent's in Melbourne, so those clinical partners have been really important. From a technology, when we've gone and looked at other applications, we've spoken with the University of Melbourne. We've had conversations with UQ as well. So we've really kind of, I guess, and internationally, but yeah, we've always kind of gone out there and, spoken with people to identify where that expertise is and how we can access it. And when before we partnered with the ACVT originally, we actually went round the world, or the CEO at the time went around the world and spoke to universities in, the US, in France, in UK, to identify the best AI partner, and then found out that we had, the best ones right on our doorstep here. So that was pretty good.
Thanks.
I think one of our challenges has been getting good access to regulatory advice. We were quite lucky, and I think we had success, but I feel that's quite a difficult bridge to cross. We found, particularly in the US, you get quite different advice if you ask someone in the US what to do, as if you ask someone from outside the US what to do in the US. I think it took us a while to realise that difference, but that's quite a challenge, I think.
Yes, thank you. Thank you for sharing openly, what made it successful and what were the challenges on its way as well. And I think it's really relevant for many people in the ecosystem. So I really like to thank you both for the time and for the presentation. For everybody listening today, it will be recorded and on the LA Biomedical website the end of this week. There are also some other recordings available of all the past webinars the last year, 1.5 years. So there are many there, many wide range of topics as well. Normally I also introduce the next week's speakers, but I forgot to look who they are. So apology for that, but just go to the website and you can find out there. Thank you again.